Reproduction in bacterium. Reproduction in bacterium. Bacterium reproduce mainly by two methods. Asexual and sexual method of reproduction. Asexual reproduction by two ways: binary fission and by endospores. First one, asexual reproduction. Asexual reproduction showed by bacterium in favorable conditions. Bacterium reproduced by binary fission. In unfavorable conditions, some bacteria reproduced by endospores. So, favorable conditions binary fission, unfavorable condition by endospores. Most common method of the replication of virus or multiplication of the bacterium. Most common method of the multiplication of the bacterium by binary fission, binary by 2. One bacterium divides into 2 bacterium. Process of binary fission. Bacterial cell, nucleoid. First, bacterium elongates. Nucleoid that divides into 2 or the main genetic material replicates and becomes the 2. Next, plasma membrane. Small invagination occur in the plasma membrane in the middle of the cell. This invagination gradually that grows centripetally. Along with cell wall also grow into that groove. Finally, 2 daughter bacteria are formed. Each bacterium consists its own genetic material. In this, one bacterium divides into 2 bacteria. Type of division, amitosis. It is the prokaryotic cell. No kinetochore in the chromosome, no spindle appendage formation, no prophase, no metaphase, no anaphase, no telophase. So, approximately bacterium cell that divides into 2 daughter cells. The type of division, amitosis, without any phases, particular phases, cell that divides into 2 daughter cells. For this division, mesosome mainly involved. Again the daughter cells they grow and again participate in another binary fission. Time taken in between 2 successive binary fissions. Time taken in between 2 successive binary fission. Doubling time. Or regeneration time. The time in between 2 successive binary fissions called doubling time or regeneration time. It is minimum 20 minutes, maximum it may be more. It is the minimum 20 minutes. So, for the division some time taken. For next division, it also takes some time. That time is called doubling time or regeneration time. Actually, this doubling time or regeneration time, that is equal to the time taken for interphase in case of the eukaryotic cell. Here interphase, such type of particular phase is absent, but anyhow any cell to divide, it must grow, and all the cell organelles or the cell substance must be grow. It takes some time. That time is called doubling time or regeneration time, that is equal to the time taken for interphase in eukaryotic cells. In unfavorable conditions, in some bacteria, protoplast that contracts, thick wall is formed, become a spore called endospore. Suppose it is a bacterium, nucleoid present, cytoplasm. In unfavorable conditions, this protoplast contracts, around that thick wall is formed, it becomes a spore called endospore. When favorable conditions return, the cell wall ruptures, endospore on germination produces the new bacterium. One endospore, one bacterium. One endospore on germinate produce one bacterium only. So it observe endospore formation, we observe in unfavorable conditions.